So this is a podium presentation on the role of protein kinase C in human myometrial contractility, which I presented at Imperial College London Young Scientists Symposium in 2006, following an Undergraduate Research Opportunities project at the University of Delaware in the USA while a Year 3 medical student. The rationale behind the study was that 1 in 12 women suffer from dystocia, meaning abnormal or difficult labor due to inappropriate uterine contractility. This cost $15.5 billion to the US healthcare in 2003, and further understanding of physiology of uterine contraction and its regulation was thought to be needed. There was a growing body of evidence that protein kinase C plays a substantial role in modulation of smooth muscle contractility, and the function of PKC in the regulation of uterine contractility is only partially understood and remains controversial. So to start off with, I looked back at the mechanism of smooth muscle contraction and relaxation, and this is a slide from a biochemistry book from 1996 demonstrating how calcium moves out of the sarcoplasmic reticulum to interact with the cell membrane, and how calcium receptors allow calcium in and out to eventually combine to interact with myosin, which then acts on actin to result in contractility. And in particular, in here, we wanted to find out or have some idea of what the different subtypes of protein kinase C do in relation to this contractility. The working model that we were using centered on protein kinase C, intracellular calcium levels, and the contractility of the muscle. So the measurements that we were making was primarily on calcium and contraction, and we looked at different ways of reducing the level of protein kinase C, or modifying or increasing PKC and modifying the levels of calcium, and measuring contraction and calcium levels. Some of the other things are I suppose somewhat extra, but we did go on to look at the role of Rho kinase, and I'll leave you to study this model in more detail if you wish. The questions that we asked: do PKC levels change in pregnancy? What are the effects of inhibiting and activating protein kinase C? Does PKC interact with Rho A to regulate contraction? But I'll leave this for today. So the PKC isoform present in human myometrium, the ones that we looked at was alpha, beta 1, beta 2 and gamma, looking at non-pregnant myometrium in black, pregnant not in labor, and pregnant in labor. And essentially some subtypes are really high or relatively higher in the contracting uterus in labor. So the flowchart essentially that we followed, starting from here, we would obtain some human or mouse myometrium, depending on the study we were doing, cut the muscle strip to a particular size, which we used as 15 by 2 millimeters, decide if we were going to change the level of PKC by modifying DNA. If yes, we would first incubate it with the appropriate nucleotide as required. If not, we would go on to measure calcium and contractility by setting the muscle in calcium solution in normal saline. We would then observe 3 contractions or 10 minutes. If we were going to add any other agents to change levels, we would add those agents, which could be oxytocin, something to increase or reduce PKC, or likewise for Rho A. And again, we didn't quite cover this in this presentation. And again, repeat the cycle, make the recordings, and stop there. So the first step was to attend the local hospital, where during a cesarean section I would obtain the sample for analysis. I would then take it back to the lab with me, set it on a petri dish, and using some instruments, cut it to obtain the strip that we would require, tie string on either side so that I could then mount it within this pot onto a tension sensor, and then obtain the recordings through the apparatus directly onto the computer, and likewise for the levels of calcium within these pots. So question 1, what happens when PKC is activated? We use an activating agent called PDBU, and we would then measure tension and calcium level. We're using pregnant human myometrium at this point. Along the x-axis is a time in minutes, so starting from 100 to 600 minutes. So these were long experiments with frequent measurements, so quite time and labor intensive. So here I would start off making the measurement before doing anything, and I could see that this myometrium with time I'll give some calcium and there will be contraction. It then drop, give some more calcium, achieve contraction, and that would correlate. I would then continue to giving the same amount of calcium but increase the activity of PKC, and this demonstrated that the tension within that muscle strip increased significantly compared to that baseline contractility before, every time I would give the same volume of or amount of calcium, and upon washing out of both the activator and calcium, this would then decrease. So then I wanted to know what happens when I activate the PKC without giving calcium. So KCl, potassium chloride, sorry, I keep using the wrong term. So this is KCl, which activates calcium, yeah, but the actual agent was called KCl, potassium chloride. So this would induce intracellular calcium, and just to confirm that the muscle works, we demonstrate contraction. We then get a washout, so calcium level stays low. But despite this low level in calcium, if I increase protein kinase C without inducing any more calcium, I obtained contraction of the muscle. So then we looked at it in some further steps. So again, test the muscle strip. Yes, when we give calcium, when we activate calcium with potassium chloride, we get an increase in calcium in red, and we also get contractility. If we then block calcium or inhibit calcium with an agent called BAPTA, we can try to use the KCl, but there is no increase in calcium. And then in the next step, we've blocked the calcium, but we activate protein kinase C, and we increase contractility despite falling calcium. If we didn't give more calcium, at this point essentially there was no change in contractility, although the calcium increased. So then what happens if we reduce protein kinase C? So this time we used an agent called GFX and tested the muscle. So yes, by giving potassium chloride to increase calcium, we got increased contractility. Let's then inhibit PKC at this point, and it seems to make no real difference in that increasing calcium gives you the same amount of contractility, although PKC is not thought to have been inhibited. So let's look at it another way. Let's give some potassium. Yes, we get our contractility. Then let's inhibit PKC, but give no further so it maintains the same level of calcium, but there are no further contractions. So no further boluses of KCl. So let's inhibit PKC and then let's activate PKC and then inhibit it. So test, standard, more calcium, more contraction. Activate PKC, increase contractility, inhibit PKC in the presence of the activator. The inhibitor is stronger, we maintain the calcium but we lose contractility. Back to the baseline at this point. So what happens if we deplete protein kinase C? And what I mean by deplete is incubating with nucleotides that contain antisense to PKC so that it's no longer made. So in the control one, give potassium, tension increases, and then we give the stimulant to increase PKC, and we've achieved increased in contractility as expected and demonstrated previously. On the other hand, if we've depleted it, we can confirm that although there is the same increase in tension, or lower increase in tension, by activating with PDBU, i.e. activating PKC, there is no further increase in contractility. It has nothing to work on essentially. So again, what happens when PKC is depleted, this time on calcium levels? So control, induce calcium, you get a contraction. So you induce calcium, you get increased calcium, and then it washes out. In the antisense, again, the calcium increases and then drops in the same pattern, and then stimulating with a PKC activator makes no real difference. I won't talk about Rho today, but in summary, PKC alpha and PKC beta are differentially expressed in uterine smooth muscle during pregnancy, with highest expression in labouring myometrium. PKC beta is not expressed in non-pregnant myometrium but expressed in pregnant uterus. Activating PKC increases tonic and phasic myometrial contractility without changes in intracellular calcium. Inhibition of PKC diminishes the effect of its activation, but inhibition alone was not found to have an effect on contractility. Depletion of PKC alpha reduces uterine contraction. In conclusion, we settled on that the role of PKC in regulation of human myometrial contractility is complex and still incompletely understood, but it appears to be involved in calcium-independent contractions. So I'd like to thank my supervisor, Dr. Duncan, the PhD students I was working with, Andres Krotzenbergs, my direct supervisor, Victor Fomin, and Dr. Zhang for assisting with some of the molecules and chemicals we used for this experiment. And I also like to thank my previous research supervisor, Prof. Openshaw, Wiesza Olszewska, Prof. Parker, and Dr. White, and my educational supervisors at Imperial, Dr. Marshall, Dr. Gentleman, and the Undergraduate Research Opportunities Coordinator at Imperial, Mr. Adrian Hogsworth.